have higher metabolic rate than the healthy cells. It means that the tumor microenvironment have much more conductivity than the normal cells around. It means that you can, if you have a good radiofrequency current through the body, a good frequency, good dose, then of course you can select for this very special.
I think as most of you are physicians, you don't need me to remind you of this, but currently the techniques of diagnosing computer-based tomography, which is very expensive and also results in radiation exposure on the patients. There is also tissue biopsy, which is very invasive and complicates.
Here I'm going to talk about the novel delivery modalities for cell engineering. So there are in vivo cell engineering and ex vivo cell engineering. So for the ex vivo cell engineering means we add the cells outside the patient and then reintroduce them. And for in vivo cell engineering, we add the cells directly in the patient. And for both cell engineering methods require
everybody to talk. So in gene therapy, certainly to address so many unmet medical needs, so many genetic disease need to have the efficient gene therapy, which can
one of the prominent cancer across the worldwide, and second prominent cancer in India. So in my previous presenter has already shown the data of the many cancer and breast cancer also. So total 2.3 million breast cancer cases have been reported in 2024 in women worldwide. Among them, 238,085 new cases reported in India, and mortality cases in India in 2024 is 1,23,077.
After that, we did the number of mitochondria in the ovaries of the aged mice and find out that in the aged mice, the numbers or the conditions of the mitochondria was reduced.
All these herbs, hey doctor, you just combine all this grass and use this all grass to the patient? No, the regular in Taiwan, we just make all these herbs and we just cook them to a liquid herb decoction. You know, you can imagine that just like a coffee, but it's a bean coffee. You want to drink that.
So we are about 22,000 employees in this hospital. It's the largest hospital in Northern Europe. It's one of the largest one in the whole Europe. So we are working a lot to the photodynamic therapy. And now, today, I'm going to talk about the photophoresis. So what is the photophoresis? Photophoresis starts with a patient, we take out part of the blood. Then we separate white blood cells from red blood cells.
What do you do by genetically modifying? You incorporate the gene to express a synthetic receptor called chimeric antigen receptor on T lymphocyte. And it directly targets, recognizes and binds to the tumor-associated antigens we are talking about in cancer context without the antigen-presenting cell and MHC involvement, which is a huge plus because some cancers, some tumors downregulate MHC expression, so which can be a problem.
Now you're able to run a smaller column and do multiple cycles. And so when you're doing a clinical trial, you may be doing about 3 lots. After that, you put your column and the resins away. But you're not going to go back and use it because by the time you get approvals to go to to phase 2 clinical trials or phase 3 or approvals for product to be launched, that could be 2 years, 4 years, 6 years. The resin would have expired. So you can get away with smaller column and do multiple. So instead of doing a product which will take you 8 to 10 hours to do the purification, now you can do it in 2 hours and do multiple cycles.
Regulators often cite imaging quality as the basis for drug approval. So this is vitally important to consider when you are conducting your clinical trials. Before we begin, just show of hands. Has imaging ever delayed your clinical trial? And if you think yes, you're not alone. In fact, imaging delays are cited as top 3 reasons for trial oncology delays. So the roadmap for today, we're gonna talk about why imaging is so vital. We're gonna talk about typical pitfalls that you may see, how to mitigate them, and establish a good governance metrics.
The tumor is actually, as you'll see in a minute, a tumor of the peripheral sympathetic nervous system. So it's not a brain tumor, it's a peripheral sympathetic tumor of the system that produces adrenaline for flight or flight from the adrenal medulla.
The tumor disappears. The tissue surrounding, the healthy tissue surrounding the tumor heals, while everyone else stays the same. So basically what you want to do is to go from the illness to complete healing everywhere in the body. So this is obviously a big problem in the field of cancer therapeutics. And in fact, if you think about the advances that we have made with non-targeted therapies like chemotherapies or radiotherapies, for example.
Studies have shown that, in the setting of cryoablation, there's release of tumor-associated antigens and also damage-associated molecular patterns. These DAMPs are, basically proteins that help activate dendritic cells, which are, antigen-presenting cells that help activate the immune response. So this combination of tumor-associated antigens and proteins that activate dendritic cells is a recipe for activation of T cells that can respond to tumor. Similar to microwave ablation, cell damage, cold damage, you're going to have a pro-inflammatory response, just your body's natural healing response. And studies have suggested that there may even be a higher pro-inflammatory response after cryoablation compared to microwave ablation.
Past few decades, target identification and prioritization have been revolutionized by the advent of functional genomics screen, from very early on RNAi-based screens all the way up to Achilles and DepthMap, which defined gene interactions that can then be exploited therapeutically. A powerful example of how such an approach can be successful is the exquisite sensitivity to P85.